well. So today we're going to start a new topic in MOL. So it's going to be about amino acids degradation. Okay, you already started this with Dr. Raja, but when Dr. Raja lecture, you focused on which part in amino acid degradation?
Amino.
Yes, the fate of the amino group. Okay. Now we're going to focus on the fate of the carbon skeleton, and we're going to see different pathways that different amino acids will go through to be metabolized. And to do that, we have 4 lectures. So we have 4 lectures covering the pathways that amino acids take to be degraded, and also related disease and products that are formed from these amino acids, etc. So today we are going to talk about the tissues where amino acids are degraded. We're going to classify amino acid as glucogenic or ketogenic. So we need to define what does glucogenic mean, what does ketogenic mean. And we're going to remind you of essential, non-essential amino acids. And we're going to introduce the inborn error of amino acid metabolism. And try to highlight the major pathways involved in amino acid degradation. So this is the overview of today's lecture. This lecture will set the stage for the future lecture. So in future lecture, we'll come back to these different pathways in more details. So today we're going to have an overview of all the pathways, but in future lecture we're going to take pathway by pathway and talk about all the details relevant to this pathway. First of all, the location. Where do amino acids get degraded?
Liver.
Yes, mainly in the liver. Yes, the liver is the principal site. There are other tissues like kidney, small intestine, muscle, and adipose tissue, but the main site is the liver. Importantly, the branched chain amino acids. Sorry, what are the branched chain amino acids? Can you remember from MOL 1?
Lysine,
Someone is saying a correct answer. Some other people are saying I'm mixing up.
Valine,
Valine, leucine, isoleucine. Valine, leucine, isoleucine. These are the branched chain amino acids, and they have branches in their R. That's why we call them branched chain. We're going to see that they share the same pathway, and there is a disease in amino acid metabolism which is related to their yes, what is it? Maple syrup. Yes, I introduced this to you briefly in MOL 1 today, or in these lectures we're going to talk about that in more detail. So these branched chain amino acids are degraded mainly outside the liver and mostly in muscles. So most amino acids usually are degraded in the liver. For the branched chains, they are degraded mostly in the muscle. So what is the fate of these amino acids? So after we remove the amino group, which you studied with Dr. Raja, what will happen to the carbon skeleton? The carbon skeleton will be further metabolized, and it can be used as a source of energy. It can be used to form glucose or ketone bodies, or it can be used to make other compounds. So this is what happened in a nutshell, what happened to the carbon skeleton after you remove the amino group. So according to their fate, so what will happen to the amino acid next will determine whether they are glucogenic or ketogenic. So this is a classification based on the fate. So we will see today 2 classification based on whether we can make them in the body or no, and the classification depending on what is their metabolic fate. Will they form glucose or will they form ketone bodies? Okay, so I'm not going to focus on this part. So this part it's already covered by Dr. Raja. My job with you is to focus on this one. So the carbon skeleton or the corresponding keto acids, sometimes we refer to it as alpha keto acid. So when you hear the word alpha keto acid, this means that we are dealing with the carbon skeleton of the amino acids. So this is to summarize what I've said in the previous slide. So the carbon skeleton can be used to make other compounds. Can you give me example of a compound which is derived from the amino acids?
Glucose.
Compound, like not energy, not glucose, not ketogenic.
Ammonia.
Glutamate.
Glutamate is an amino acid, right?
CO2.
Serotonin, yes. So serotonin is derived from amino acid. Dopamine is derived from amino acids. Okay?
Epinephrine, norepinephrine.
We're going to talk about this in more detail. So this is the image that should be clear in your mind, that carbon skeleton can be used to make other compounds that the body needs, or it can be further catabolized to produce energy. Come on, under this umbrella I would see glucose and ketone bodies, because the ultimate goal of glucose and ketone bodies is to produce energy. Again, you took this part in the previous lecture, so today we're going to focus on the metabolism of the carbon skeleton, and we're going to see that these 20 amino acids that make up our protein will eventually be broken down to one of these 7 intermediates. And accordingly, we're going to classify the amino acids to ketogenic or glycogenic. So those who give, once they are metabolized, give rise to acetoacetate or acetyl-CoA or acetoacetyl-CoA, we're going to classify them as ketogenic. Why? Because these are ketone bodies. They will provide the body with lipids and energy. And those who will, once metabolized, those amino acids who are metabolized into one of these 5 will be classified as glucogenic, because they're going to give rise to glucose. Yes, they can give rise to lipids as well, but the main thing is that they give rise to glucose. Type. What is common among these 5? Yes, so these 4 are intermediates of the Krebs cycle. Pyruvate is the end product of glycolysis, and it can be used to feed into what? Which reaction? Gluconeogenesis. So all these, what's common among all of them, and all these 5 compounds can be converted to glucose. Okay, so all of these can be converted to glucose. How? Pyruvate, you study the gluconeogenic reaction, oxaloacetate the same, and these 3, how they can be converted to glucose? Yes, they can give rise to oxaloacetate, and then oxaloacetate can produce glucose. So this is the classification based on the fate of the amino acids. So if they are metabolized to give rise to acetyl-CoA, acetoacetyl-CoA, we'll call them ketogenic. If they give rise to pyruvate or one of the intermediate of the Krebs cycle, we're going to call them glucogenic. So they are able to give me glucose. These, they can't give glucose, they only can give lipids and ketones. Okay, by looking at here, so this is the metabolic fate of the 20 amino acids. Can you give me an example of an amino acid which is glucogenic?
Aspartate.
Yeah, so that's the easy one. Alanine is glucogenic, can give rise to pyruvate. Okay, someone mentioned aspartate. Yes, aspartate can give rise to oxaloacetate. So this is how it works. So if they give rise to pyruvate or one of the intermediate of the Krebs cycle which can be converted to glucose, we're going to call them glucogenic. So in red, what do we have here in red? The amino acid labeled in red are the one? Glucogenic. The ketogenic are labeled in green. So how many green we have? 2. Lysine and lysine, leucine and lysine. So these will give rise to Acetyl-CoA. So acetyl-CoA can be converted to fatty acids or ketones, right? طيب, what about the purple?
Both.
Okay, what do you mean by both? Give me an example. Tyrosine. Let's look at tyrosine. So tyrosine is here because its degradation will produce acetyl-CoA, and tyrosine is here as well because its degradation will produce fumarate. Okay, so tyrosine metabolism will produce both acetyl-CoA and fumarate. As a result, what is the classification of tyrosine?
Both glucogenic.
Yes, gluco and ketogenic. And this applies to all the amino acids in purple. And these are summarized in here. Okay, so it will be easy to remember that the two amino acids that start with L are ketogenic. So KL, the order of the alphabet. For the both, okay, let's try to find a way to remember them. What's common among three of them?
Starts with T. Anything that starts with T is both.
Anything that starts with T? Okay, one way. طيب, phenylalanine, tyrosine, tryptophan, what are they?
Aromatic.
The aromatic. Okay, so the aromatic amino acids are all both ketogenic and glucogenic, which makes sense because they have a bulky structure, a big structure, so it's more likely that they can give rise to both acetyl-CoA and another intermediate, which is in the Krebs cycle. طيب. In addition to the aromatic, what do we have? We have isoleucine. Okay, so this is the branched chain amino acid, and you have another T, which is threonine. Okay, so all the T's, if you want, in addition to phenylalanine, which is another aromatic, and isoleucine, which is one of the branched chain amino acids. So and this is both gluco and ketogenic, and the rest is glucogenic. So we learn from this that most of the amino acids, except two, will give rise to glucose. That's why, in time of hunger, in time of fast, we need those amino acids as a source of glucose. That's why, in the diet they always talk to you about muscle wasting, because the amino acids can give rise to glucose if you don't have enough energy in your body. We covered this in Mol 1. Can you remind me of essential and non-essential?
You can make it.
Okay, which one can be made in our body? Non-essential. So we're going to see synthesis reaction for the non-essential. There will be reaction for those because the body can make them. The essential, I always tell my students, how do we remember essential, non-essential? Essential? No, remember how to remember essential, non-essential. Essential to eat, you have to eat them. Your body can't make them. Okay, so those who must be obtained from the diet are going to be essential. So must be obtained from the diet, essential to eat. You have to eat them. This is the easiest way to remember them. And you will see reactions for the non-essential. There is something called, in some books, or previously they used to call it semi-essential, now they call it conditionally essential. What do we mean conditionally essential?
Sometimes the body doesn't
need it
make
at
it.
some stages. The synthesis is not that much.
The body cannot synthesize it.
Yeah. No, the body can make it. There is a reaction to make it.
Yeah, but it doesn't.
It doesn't make enough, yes. So, under certain condition, the endogenous production of this amino acid is not enough. So, you have to get it in the diet. So, during period of growth or positive nitrogen balance. So, when the body is healing itself, when during pregnancy, etc. So during period of when there is like pressure on the body to have lots of arginine. The arginine that is made in the body is not enough, therefore it will become conditionally essential. But for the sake of avoiding confusion, it is included here. So you have the acronym PVT, Tim Hall, to remember the essential amino acid, and acronym NANOE. Next, we're going to talk about inborn error of amino acid metabolism. So these are the same as, I'm going to refer them as metabolic defect in amino acid metabolism. So first of all, let us define the terminology. What do I mean by inborn error? Something that is present at birth. Okay, so it is an inherited defect. So inborn error of amino acid metabolism. So it is a defect that is present at birth in amino acid metabolic pathway. Most of these diseases are going to show up during the newborn period. We'll see like rarely one or two that will show up later in life, but most of them, they will show up during the newborn period. And the clinical features are non-specific. So you're not going to see like glycogen storage disease, you're not going to see hepatomegaly, etc. So what will happen? What are the presentation? You will see that the baby is not growing well, so failure to thrive. He's not putting on weight properly. He has hypotonia. What do we mean by hypotonia? Poor muscle tone. And he will have signs of developmental delay. So he will have symptoms in the central nervous system. So how they are diagnosed? By laboratory testing. And when do you suspect inborn error of amino acid metabolism? Mostly if there is no newborn screening. What do we mean by newborn screening?
Genetic testing.
You took it in genetics?
Screening after birth.
Yes. So let me explain it on this slide. So newborn screening is when you test a newborn shortly after birth for serious but treatable disorders. Treatable doesn't mean that there is a cure, but you can manage them. And usually, for amino acid metabolism, you have to give them a specific diet. Like you avoid that amino acids that they cannot metabolize. It should be available worldwide, but sadly it's not. So underdeveloped countries, like some countries in Africa, they don't have newborn screening. In Saudi Arabia, it was made mandatory in recent years, I think in the 90s, and it's usually performed at 24 to 72 hours after birth. And besides, screening for metabolic disease, so it's not only amino acid metabolism error that they will screen for, but they screen for other metabolic disease, like I don't know, in Saudi Arabia it includes several screenings. So besides phenylketonuria, they screen for I think organic acid metabolic disease, etc. Endocrine disease and blood disorders like sickle cell anemia. So the screening is not only for amino acid metabolism disorder, but also for other inherited disease. And this is important because of the high consanguinity in Saudi Arabia. You know what's consanguinity, right? You took it in genetics. So because of the presence of high consanguinity, you will find that inherited disorders are common, such as sickle cell and other disease. So for this lecture, you will see that also PKU, phenylketonuria, has the higher rate in Saudi Arabia in comparison to other countries. What do you think of this slide? Easy. Okay, so this is what we are going to cover in the next few lectures, 4 lectures. So this is the metabolism of amino acid. So what happened to every amino acid, how it is metabolized, and the disease. So these X means that there is a inherited disorder. So we're going to talk about maple syrup, phenylketonuria, alkaptonuria. You already took Hanaa and pellagra, right, with Dr. Raja? And we're going to talk about the synthesis of other products from the amino acids. So this is the full picture that we will see, you know. So 4 lectures, you already took 3 lectures with Dr. Raja, so this is not 1 lecture. I think I skipped. Yeah. So when we talk about inborn error, we have to appreciate why it's important to do the neonatal screening or newborn screening. There is a simple reason why it's important to do it soon after birth. And by reading this slide, you will understand why it's important to do newborn screening. Why don't we leave it later? Like 2 weeks. When they come back for the vaccine, we screen them for the newborn error or inborn error of amino acid metabolism and other metabolism disorder. Why do we have to do it soon after birth?
Because they're going to start eating or drinking, and that might have effects on their metabolism.
Okay. طيب. We will treat the problem later. Yes.
The disease manifests pretty quickly.
Okay, so the disease manifests pretty quickly, yes. And what he said is correct. Like they will start eating, and what will happen?
They're going to start metabolizing or not metabolizing.
Okay, and what will happen if they can't metabolize something? What will happen?
AKU.
Accumulation of?
Amino acids.
There will be accumulation of metabolites. Okay, so when there is usually in biochemistry, when one pathway or one step is blocked, what will happen? Not only accumulation, but things can, will back up and start producing something harmful. So that's why you want to screen them. Are we all following? I think you're following the news. You all have your head in your screens. You reading the news? Are you worried about Trump? Let's worry about amino acids today, okay? Okay, so there will be, this is the key, there will be accumulation of substrate and deficiency of product. So this will result in problems for the central nervous system. So it can be harmful for the central nervous system if you deprive the CNS from a certain product, or you have accumulation, like we'll see accumulation of phenylketones is very harmful for the central nervous system. So you don't want, accumulation of harmful products. So you want to intervene as early as possible by giving that baby the right diet. So usually they can give them a special baby formula that is deficient in the amino acid that they can't metabolize, and therefore they prevent the irreversible damage to the central nervous system. So if detected early, they can prevent, formation of harmful metabolites which can damage the central nervous system. That's why it's very important to screen early for these metabolic defects. Okay, for the rest of today's lecture, I'm going to go through these different pathways. So this is the list of pathways that we'll see. It's an introduction. We'll come back to them in details when there is a disease related to them or when there is a product that is derived from them. But for today, we're going to introduce those 7 pathways, and we're going to talk about the major products that are formed in this pathway. You will see that certain amino acids, they share some pathway. So for instance, phenylalanine is converted to tyrosine. Okay, so by default, it's going to be, a one pathway. Similarly, threonine and serine, they get converted to glycine. So that's why they share a pathway. Methionine is converted to cysteine. Histidine is on its own, it doesn't share pathways with another thing. The branched chain amino acids, so the 3 branched chain amino acids, they share the same pathway. And some of them, like valine and isoleucine, they also share what we call the propionyl-CoA pathway. You took that where? Propionyl-CoA pathway? Yeah, with Dr. Abdul Jabbar. Okay, so we're going to go through these pathways one by one. So we're going to start with the simplest, which you already took with Dr. Raja. Some amino acids are simply degraded by removal of the amino group. There are no multiple steps. You remove the amino group, you have a corresponding keto acid, which is pyruvate or oxaloacetate, or it's mainly pyruvate or oxaloacetate. Or you can, if you remove the amino group from glutamine, what do you get? Glutamate. And from asparagine, what do you get? Aspartate. Okay, so these amino acids will give me the, one of these 7 products. Remember, so these 7 products which can be either converted to glucose or to ketones. So in that case, glucose. So these 3 here, and this one, aspartate itself, can be converted to oxaloacetate. Glutamate can be converted to alpha-ketoglutarate. So these 5 amino acids, just you remove the amino group. There are no multiple steps. You just remove the amino group, and you've seen those enzymes. What are ALT, AST? Yeah, and you took them in many lectures. And which other lecture? You took them early in the course? Liver enzymes. But no. When you measure the liver function, 2 of the important enzymes that you measure for liver function is ALT, AST. So conversion of amino acid by removal of the amino group. So this applied to all of these. You just remove the amino group from alanine to get pyruvate, from aspartate to get oxaloacetate, etc. For asparagine, you remove the amino group and you get aspartate. To do that, you need asparaginase, and I'm going to discuss it in the next slide. Glutamine, also you remove the amino group by glutaminase and you get glutamate. So this is a revision of the ALT and AST. I'm not going to go through it again. You have studied them multiple times. Let us focus next on asparaginase. So what does asparaginase do? It removes the amino group from asparagine to produce aspartate. So why here I have it, because it has clinical application. What is the clinical application of asparaginase? Well, it turns out that normal cells can make asparagine, but leukemic cells, they need lots of asparagine, and they can't make enough asparagine. So leukemic cells depend on circulating asparagine, and if you deplete that circulating asparagine, that will lead to the death of the leukemic cells. So that's why asparaginase enzyme is used to treat ALL. What is ALL? So it's a type of leukemia, acute lymphoid leukemia. Okay, so this is asparaginase enzyme, which is used to treat leukemia, because leukemic cells need lots of asparagine, and by depriving them from asparagine, you kill them. Okay, so we're done with the first easy pathway. Next, we're going to introduce phenylalanine and tyrosine metabolism. So phenylalanine is essential. There are no reactions to make phenylalanine, but phenylalanine gets converted to tyrosine. So does this make tyrosine essential or non-essential? Saying that phenylalanine is converted to tyrosine, so it means that you have a reaction in your body to make tyrosine that will make tyrosine non-essential.
But don't you need phenylalanine essential? So
No.
indirectly it should see.
Because you have a reaction to make tyrosine, that's make tyrosine non-essential. And some textbook they call it as semi-essential, but this is not correct. Because you have a reaction, then it is non-essential. So how does phenylalanine get converted to tyrosine? By hydroxylation. So we need to add what? OH. So that means that tyrosine is phenylalanine plus an OH group. And to do that, we need a special cofactor, not tetrahydrofolate this time. What is it called? Tetrahydrobiopterin. So this is BH4 or tetrahydrobiopterin. We'll come back to it in details. Don't worry about the full name now. But BH4 is the cofactor to convert phenylalanine to tyrosine. And we will see later that tyrosine is the precursor for many important molecules such as the catecholamines, melanin, thyroid hormones. Okay, and this is summarized in here. So phenylalanine gets converted to tyrosine, and the enzyme is, what is the enzyme? Phenylalanine hydroxylase. So we're going to add the OH group, and this is the cofactor, which is BH4 or tetrahydrobiopterin. Once we form tyrosine, you will see that in the metabolism of tyrosine, we will form something called homogeneity. We'll come back to this later on. And eventually, tyrosine is broken down to fumarate and acetoacetate. That's why tyrosine is classified as,
Both ketogenic.
yes, both glucogenic and ketogenic. So this is how phenylalanine and tyrosine, they share one metabolic pathway, and we'll see the disease later on, the deficiency of phenylalanine hydroxylase, and we'll see a disease which is the accumulation of homogeneity, and both are the types of inborn error of amino acid metabolism. Next we have threonine, serine, and glycine. Why they share same pathway? If you look at it here, you will see that threonine can be broken down to pyruvate, succinyl-CoA, but also acetyl-CoA. But threonine can be converted to glycine. Okay, first of all, what is threonine? Glucogenic, ketogenic, or both? Both, because it produces acetyl-CoA and pyruvate. Threonine gets converted to glycine, but also serine gets converted to glycine. Glycine, which is coming from threonine and serine, has two fates. It can either be completely metabolized to form CO2 and ammonia, or it can be converted to glyoxylate to give rise to oxalate. Where is oxalate present? You took it in a previous block. No, block, not this course.
Kidney?
Yes, the stones, kidney stones are formed of calcium oxalate, and the oxalate is coming from glycine. Okay, so there are two fates for glycine. The major pathway for glycine degradation is this is glycine here. So we completely break it down into CO2 and ammonia, and the carbon here, the remaining carbon, is donated to tetrahydrofolate to form methylene tetrahydrofolate. Okay, so we call it, it's going to generate the one carbon unit. So it is a donor of the methyl group for methylene, for tetrahydrofolate. So this is another way of writing the TH4. Okay, tetrahydrofolate, so it's folic acid, and H with 4 hydroxy group or 4 hydrogen, and it can be converted to methylene tetrahydrofolate by taking the carbon. So this carbon can be taken by tetrahydrofolate, and it will become methylene tetrahydrofolate. So this is the major fate of glycine, and we call this reaction glycine cleavage reaction. So this is glycine cleavage reaction. So there's complete cleavage of glycine into CO2, ammonia, and the other carbon is donated to tetrahydrofolate. The other fate for glycine is to form a glyoxylate, which can be converted to oxalate, and it is the oxalate which is responsible for the formation of calcium oxalate crystals in urine, or the kidney stones, which is the major type of kidney stones. Okay, so we're done with threonine, serine, and glycine. Next, we have methionine and cysteine. This is a slightly more complicated pathway. We will come back to it in more detail, but no, for now, let us have an overview of it. So in the metabolism of methionine, we're going to meet a very reactive molecule called SAM. What is SAM? It is S-adenosine methionine. So it is methionine plus what? Okay, so we're going to take everything from ATP? No, we're going to take the adenosine from ATP and add it to methionine. So you can see here, there's no phosphate. We just have the sugar and the ribose, right? So we have ribose sugar, sorry, we have the base and the ribose. I said sugar and ribose, right? Okay, so this is the sugar, ribose, and this is the base. The base is adenine, and this is the adenosine, which is a nucleoside. If you remember from mole 1, without phosphate, we call it a nucleoside. So this part is coming from ATP, and it is added to the S of methionine. So we call it SAM because it is S, because we have the adenosine attached to the S of methionine. And this S here is very reactive. So it wants to donate this adenosine, and if it donates this adenosine, it will become SH, which is a harmful, which is homocysteine, and it is very harmful. So we'll explain this. So SAM is the methyl group donor in the body. Is actually the major methyl group donor. We'll see that it donates the methyl group to epinephrine to form norepinephrine. So SAM donates its methyl group and it will become SAH, which is S-adenosyl homocysteine, which will hydrolyze into homocysteine, and homocysteine is very harmful for the body. Okay, that's why the body will try to get rid of homocysteine, and there are two fates. Let me explain it in here, it might be easier. So this is methionine. Methionine will get activated by taking the adenosine from ATP, so it will become S-adenosyl methionine. So it's basically methionine plus an adenosine group, and then this SAM tend to donate its methyl group here in blue, so it will form methylated product. Once it donates its methyl group, it will become SAH, and this SH will hydrolyze to form homocysteine. Homocysteine is very harmful. The body will try to get rid of it, and there are two fates for it, two possible ways of getting rid of homocysteine, either by returning the methyl group to it, remethylation to form methionine depending on the need of the cells, and to do that it requires B12, or we can do a transsulfuration. So we want to transfer this sulfur group and with the addition of serine to form what we call cysteine, the amino acid cysteine. So either we remethylate methionine or to regenerate methionine, so we add back the methyl group to this homocysteine to form methionine, or we transfer the sulfur to form cysteine. So we call it this transsulfuration because we're going to take the sulfur group from homocysteine, add to it serine to form the amino acid cysteine. That's why cysteine is formed from methionine. But to do this part here, we need B6. So in either case, we need enough vitamin B For this one, the remethylation, we need enough B12, and for the formation of cysteine, we need enough B6. Okay, and this is summarized in here. So this is to summarize how we form cysteine. So cysteine is formed by taking the sulfur group from methionine and the carbon skeleton of serine, and this is how we form cysteine. So is cysteine essential or non-essential?
Non-essential.
Non-essential. How is cysteine catabolized? It will generate inorganic sulfide, which will form sulfate, which will be excreted in the urine. So we get sulfate because, you know, the methionine and cysteine, they are the only two sulfur-containing amino acids. So their metabolism will eventually produce sulfate, which is excreted in the urine. This is the only amino acid that will produce sulfate. Next, we're going to talk about histidine metabolism. Okay, histidine metabolism. So histidine gets converted to alpha-ketoglutarate. I don't expect you to remember all these steps, but you should remember that in the metabolism of histidine, we form a very important product, which is FIGLU. What is FIGLU? It is formimino-glutamate. So what is formimino-glutamate? It is something that will be used to form formimino tetrahydrofolate in the metabolism of histidine. So what is the clinical significance of this FIGLU? FIGLU will accumulate in folate deficiency. Why? Because THF or folic acid is required to accept the formimino group. If we don't have enough folic acid, then FIGLU will accumulate. So it is a test for folic acid deficiency. Okay, so the FIGLU excretion test is a marker for folate deficiency. We've seen here one form of, tetrahydrofolate, but we'll see other forms. So one form is in the remethylation of homocysteine. We'll come back to it, the N5-methyl tetrahydrofolate. And in the later lecture, in nucleotide metabolism, we're going to see these two, the N10-formyl tetrahydrofolate, which is in purine synthesis, and in the DTMP synthesis, which is thymidine nucleotide, we're going to see N5 and 10 methylene tetrahydrofolate. So lots of tetrahydrofolate derivative to look for beside this one here, which is the formimino tetrahydrofolate. So there are 3 more to look for. Okay, you've seen in your blood block that folate deficiency can cause megaloblastic anemia. Okay, next we're going to talk about branched chain amino acid metabolism. So what are the branched chain amino acid? Valine, leucine, isoleucine. Okay, they are first metabolized to alpha keto acids. So we remove the amino group and you get alpha keto acids. So transamination reaction will donate the amino group to alpha ketoglutarate. Alpha ketoglutarate will become glutamate, and these amino acids will become alpha keto acids. And then they meet a very important enzyme, which is the branched chain keto acid dehydrogenase.
The 5 coenzyme.
Yes, excellent. So this is the last enzyme in biochemistry that you will see that require the 5 coenzymes, and this is the one that is deficient in the maple syrup urine disease. As you see, so these 3 amino acids will be eventually metabolized to acetyl-CoA and also succinyl-CoA. All of them?
No.
Leucine, not leucine.
Leucine will only give rise to acetyl-CoA, so it is exclusively ketogenic.
Also valine.
Isoleucine will give rise to both, but valine will give rise only to succinyl-CoA. So we have a glucogenic, keto and glucogenic, and ketogenic. Okay, so we said that they require the 5 coenzyme, which are the lovely coenzyme for nerds, and we've seen them in previous lectures in Mol 1. So we've seen that pyruvate dehydrogenase require these enzymes, alpha ketoglutarate dehydrogenase require those enzymes, and the third enzyme is the branched chain keto acid dehydrogenase. require also these 5 coenzymes. And you should remember, so that you don't tell me, oh, this is in mol 1, TPP is derived from thiamine or vitamin B1, lipoic acid is not derived from vitamin CoA, is derived from B5, FAD is derived from B2, NAD is derived from B3. Last pathway for today, the propionyl-CoA pathway. And this pathway will see that valine, isoleucine, so 2 of the branched chain amino acid, but in addition, methionine and threonine, they all converge into this pathway. So they will all produce eventually propionyl-CoA. Okay, and propionyl-CoA will be converted to succinyl-CoA, which will enter the Krebs cycle. This pathway will require 2 vitamins, B7, which is biotin for the propionyl-CoA carboxylase, and B12 for methylmalonyl-CoA mutase. We'll come back to these 2 enzymes later on. So if there is a deficiency of B12, methylmalonic acid will accumulate, and this is the reaction. So methionine, threonine, isoleucine, valine, they all eventually form propionyl-CoA. So this propionyl-CoA will be converted to succinyl-CoA, which will enter the Krebs cycle. So that's why they share the same pathway: methionine, threonine, isoleucine, valine. Okay, so this is a summary of what we have studied today, and this is the end of my lecture.